Good evening, is this available? Yes it is. Please leave me alone, we are sleeping. No more contacting, please. Thanks, appreciate you contacting me. I know I'm no longer interested. Please stop contacting me now. I will contact Attorney General if you do not stop the sex.
Hello. So that TikTok video was an example of allosteric regulation. And if you're familiar with TikTok, that video will probably make a little more sense for you. And if you're not, this presentation will hopefully wrap things up in a little bit better way so you understand at the end. So there are two main types of allosteric regulation. Allosteric activation. This is when an allosteric effector known as an activator binds to the allosteric site and promotes enzyme activity in order to convert a substrate binded to the active site to a product. And then there's allosteric inhibition. This is when an allosteric effector known as an inhibitor binds to the allosteric site and prevents binding of the substrate to the active site so it cannot be converted to product. Both of these types of allosteric regulation have to do with the feedback inhibition and precursor activation pathways. Feedback inhibition is when the product produced by the enzymes works their way back to the beginning of the pathway, binds to the allosteric site, and acts as an allosteric inhibitor to stop product formation. And then precursor activation pathway is when an initial substrate binds to the allosteric site, activates the pathway in order to produce more products. So today we're going to be using the glycolysis pathway as an example because it is a great example of both types of allosteric regulation. As we should know by now that glycolysis is the conversion of glucose into pyruvate while producing ATP for energy use. There's a lot more that goes on, but we're only going to talk about 2 steps in it today just to cover our bases for allosteric regulation. So the first site in the pathway that we're going to talk about is the conversion of fructose 6-phosphate into fructose 1,6-biphosphate by the hydrolysis of ATP in the pyruvate fructokinase enzyme. So during fermentation, when oxygen is absent, the aerobic pathway does not produce enough ATP and energy levels are low. When ATP levels are low, ATP will bind to the active site of the PFK, or the pyruvate fructokinase enzyme. ATP will be hydrolyzed, and the phosphate group is added and creates the product fructose 1,6-biphosphate. This is what's normally happening in the glycolysis pathway. But when energy levels are high because of the production of ATP from the aerobic respiration pathway, ATP will bind to the allosteric site. The enzyme will change the conformation and prevent the binding of other ATP molecules to the active site. ATP here acts as an allosteric inhibitor and stops the production of the product, fructose 1,6-biphosphate. So the whole glycolysis pathway stops here. This is a great example of the feedback inhibition pathway because of the product of the aerobic respiration pathway, ATP, is coming back and stopping the reaction of the enzyme, inhibiting glycolysis. A second regulation site is the next stop when the product of the last step, fructose 1,6-biphosphate, is used to create ATP by the enzyme pyruvate kinase. A substrate phosphoenol pyruvate binds to one of the active sites of pyruvate kinase, and ADP binds to the second active site. Fructose 1,6-biphosphate binds to the allosteric site, changes the conformation of the enzyme, and the two substrates are brought together. The phosphate group from the phosphoenol pyruvate is moved to the ADP and creates ATP. In this case, fructose 1,6-biphosphate becomes an allosteric activator, activating the pathway and allowing the production of more product. So those were 2 examples of both the allosteric activation and allosteric inhibition. We can apply this to the real world in multiple ways. I have 3 listed here. There's many more, but we're only going to talk about these 3. So the one being pharmacology, understanding how our bodies regulate certain pathways can contribute to the creation of medications that can either inhibit or activate certain pathways for our benefit. Second being cell signaling, the ability of cells to communicate with one another by the number of products or substrates around them, such as quorum sensing. It's a highly studied field. And then lastly is another example would be agriculture. The agriculture industry is extremely important when it comes to the production of healthy, quality, and a lot of food for the world. Understanding the regulation of these cells within plants and animals is vital for the industry and us.